Good evening. Thank you for joining us tonight for our RiverWatch Winter Speaker Series. For those of you who are not familiar with Illinois RiverWatch, we are a community science organization. We train volunteers to monitor water quality in their own communities. The mission of RiverWatch is to safeguard the future of Illinois rivers and streams through stewardship, education, and sound science. Before we get started, I would ask that you keep your microphones muted during the presentation. We will have a Q&A session at the end of the presentation. So if you do have questions along the way, you can either put it in the chat box to be answered at the end, or you can save your questions and ask them aloud at that time. Now I would like to welcome our presenter tonight, Dr. Bob Weck. Bob has been a biology professor for 32 years, and for the last 15 years, he has been researching aquatic invertebrate communities in Southwestern Illinois caves. Bob serves on the board of directors for Clifftop and the Illinois Speleological Survey. He and his wife, Nancy, are the owners of Stemler Cave Nature Preserve. And with that, I would like to welcome Bob Weck. Thank you for coming tonight, and I look forward to hearing all about cave snails.
All right, we got it. Okay, folks, first of all, I want to thank the RiverWatch program for the invitation to speak. For all of you out there who know anything about RiverWatch, it is just an excellent citizen science program. We have actually integrated aspects of the RiverWatch program into our curriculum at SWIC. And NGREC has actually supported my research through their summer internship program. So I was fortunate enough to have 3 SWIC students receive summer internships through NGREC to participate in cave research and carry out some research projects that I designed. So I'm going to begin my talk tonight by talking in general terms about caves and then get into the biology. This is a map showing cave density in Illinois. The map was produced by Chuck Sutherland, who is a geologist in Tennessee, and the data came from the Illinois Speleological Survey. So the ISS is a not-for-profit group that is dedicated to exploring caves and documenting caves, and we maintain a cave database. And when you check out this map, you can see there are definitely some areas where cave density is much higher than others. There are 2 areas here in Southwestern Illinois and down in the Ozarks. So there's a surprising number of caves in Illinois. We have 738 caves in our database, and over 300 of them are in Southwestern Illinois. So when we look at these 2 cave-rich areas in Illinois, what they have in common is they are both part of the Ozark Natural Division. Illinois is divided into 15 natural divisions based on geology, biology, soil type, and other factors. And the Ozark Division is among the most biodiverse part of the state, and again is the most cave-rich part of Illinois. So it's Ozark not just in sort of name, but this area is actually an extension of the Ozarks that we find in Missouri, Arkansas, and parts of Kansas and Oklahoma. So we are at the eastern edge of the Salem Plateau, this big geological feature that creates most of the Ozark Highlands. Here in Southwestern Illinois, our caves are found within a region we call the sinkhole plain karst. Right? Karst is a geological term for areas that contain caves and other springs and sinkholes. So the sinkhole plain karst is so named because it has among the highest density of sinkholes anywhere in the country. This is an aerial map of the Stemler Cave area. So we can see the incredible high density of sinkholes in this photograph. All those circular depressions are sinkholes. And down there toward the bottom, if you see that blue circle that just popped up, that's actually my homestead. You can see the roof of my house and my barn there, and that's where Stemler Cave is located. So that's in St. Clair County. Monroe County has really the highest density of sinkholes. The Monroe County website has a great GIS tool, and you can look at sinkhole densities, and every one of those dots is a sinkhole. And many of those sinkholes are connected to caves. This is a nice graphic that shows sinkhole formation. So sinkholes form when there are pre-existing voids in the bedrock. So the bedrock we have in Southwestern Illinois in the karst areas is St. Louis limestone. So it's a soluble limestone. Erosion and both chemical and mechanical forces create these voids. Acidic water filtering through soil dissolves the limestone, and mechanical forces like water, especially with suspended sediments, eat away at the rock and create voids. We have on top of that rock then a layer of very erodible soil. So over time, the soil makes its way into the voids in the rock, and that creates these depressions we call sinkholes. So these sinkholes, I'm spending so much time talking about them because in our area in Southwestern Illinois, these are really important conduits and points of connectivity between the surface and the subsurface. This is where most of the nutrients are introduced into caves. As you probably know, caves are dark. There's no photosynthesis. They're very low energy environments. So these sinkholes represent important, again, conduits for the importation of nutrients that sustain and allow the biological community in the caves to thrive. All right, so in Southwestern Illinois, the sinkhole plain karst is really divided into 3 sub-regions. From north to south, we have the Columbia karst, which includes portions of St. Clair and Monroe County. That's around the town of Columbia. We have the Waterloo karst in the middle and the Renault karst in the south. So tonight I'm going to be talking about the diversity of cave-dwelling snails from 2 of those regions, the Columbia karst and the Renault karst. Now, the caves in our area take on a lot of different forms. Some of them many of them are pits, right? So these are vertical caves, or at least the entrance is a vertical pit. And that obviously requires a special skill set to navigate safely. You have to have skills on rope to explore these vertical caves. Most of the caves are horizontal, right? A lot of them are low and muddy and crawly, and even for cavers are challenged to navigate. Some of the caves are huge, right? And the ones with these large perennial streams, those are the habitats that have the highest biodiversity and density of cave-dwelling snails, and those are the kind of caves I like to study. All right, so again, there are over 300 caves in Southwestern Illinois, and there are a few of them that are particularly important from a biological perspective, and there are 3 caves that will sort of be the focus of the rest of my talk tonight. So these caves include Fogelpole Cave in Monroe County, Illinois Caverns in Monroe County, and Stemler Cave in St. Clair County. So we're going to start off with Fogelpole. This is Fogelpole Cave. It is Illinois' largest cave. It has over 15 miles of mapped passage. It's huge. It has a perennial stream that flows through it. Here's another view of Fogelpole Cave. For scale, you can see the cavers in the distance. And it's that perennial stream which is home to the aquatic snails that are the subject of some of my research. I serve on the board of directors of Clifftop, and Clifftop is a conservation group in Southwestern Illinois and a land trust. And we actually own a big chunk of Focal Pole Cave. We have a nature preserve called the Paul Whiteman Subterranean Nature Preserve. You can find that south of Waterloo off Kaskaskia Road. There is, so our property boundaries are in yellow on this map, and the white branching lines is an overlay of the cave passage. So there's a lot of Focal Pole Cave passage beneath the surface of our clifftop property. You can visit the site and walk on the surface at least and explore what that karst landscape looks like. So not only is Focal Pole Cave Illinois' biggest cave, but it is also the most biodiverse cave. This is a table from a 2003 study of subterranean biodiversity in the sinkhole plain karst, and Focal Pole Cave ranked #1 in diversity. There are 17 species there that are considered globally rare, and there are 14 species classified as troglobites. So troglobites are organisms that are completely limited to cave and have adaptations to cave environments that would include often reduced or absent eyes, lack of pigmentation, elongated sensory structures like antennae. So 14 troglobitic species. Now, since I'm interested in aquatic organisms, particularly snails, we would call the aquatic troglobites stigobites. So I'm interested in stigobitic caves, or at least cave dwelling snails. All right, so in Focal Pole Cave, the snails are in the group Physidae. So for you river watchers, when you're keying out your invertebrates you collect and you're monitoring, you might remember the left-handed versus right-handed snails. If you hold the snail with the opening facing you and the apex pointing up, the shell would coil and open to your left. So these left-handed snails are pulmonate snails. That means they can breathe air, so they can absorb oxygen through the skin as well as breathe air, and that makes them fairly tolerant to pollution. So on the surface, physids are known to inhabit both polluted and unpolluted waters. These snails are hermaphrodites, which means they have both male and female reproductive parts. And not only are they hermaphroditic, but they will actually fertilize their own eggs. So potentially a single physid snail could establish a new population. They're pretty prolific. They lay eggs in masses. Some of these physids are sort of nuisance animals in the aquarium industry. In Focal Pole Cave, the genus is Phycella or Physa, depending on the author. Physid taxonomy is actually kind of messy, and there's a lot of disagreement within malacologists about Physid taxonomy. As far as we can tell, the species is closely related to Physidarena. When we go back and look at the literature about the snail and about this cave, the previous studies report that the snails are small and they lack pigmentation, and they are likely represent troglobitic species. Now, the first sort of important discovery that my colleagues and I made in studying Focal Pole Cave is it's a much more complicated story than that. Not only are there depigmented or albino snails present, but there's a whole spectrum of pigmentation. This seems to be a polymorphic population that varies from pigmented to depigmented. There are at least 2 shell morphs, clear and sort of a tan colored shell. So this is a nice collage of the showing you the spectrum of diversity and pigmentation in the cave. This nice photo essay was created by Justin Elbin at the St. Louis Zoo. So one of the things we wanted to learn about this population then is what's the makeup of albino versus pigmented. So starting in 2014, we have been doing periodic census of snails. And the very first census we did yielded this data. 67% of the population were albino, and about 32 of the population were pigmented. So our hypothesis is that what we have here is essentially a cave-adapted albinistic population with pigmented snails that make their way in from the surface and likely interbreed with the albino snails that are native to the cave. Luckily, these snails lend themselves pretty well to laboratory research. I brought the snails back to the lab at Southwestern Illinois College and established some true breeding colonies of pigmented and albino morphs. So here's a photo of the lab strains of the albino at top and the pigmented form at the bottom. So by having a lab colony of snails, it allows us to design experiments in the lab to test various aspects of the snail's biology. These are some of my students over the years. We've done a number of different experiments about growth rates under different conditions, their behavior when exposed to light, and a test to see whether or not the albino and the pigmented snails really interbreed. So one of our more significant studies involved setting up experiments to cross the albino and the pigmented form. If you think back to your high school biology and remember about Gregor Mendel and his pea experiments, so we basically repeated that with snails. So we took our true breeding albino snails and combined them as juveniles with the pigmented and gave them an opportunity to interbreed, and then isolated the albinos and collected their offspring. And we found that all of their offspring were pigmented, which suggested to us that the pigmented condition is a dominant trait and the albinism is recessive. And then we allowed the F1 snails to interbreed to create a second generation. And you can see in the lower part of the screen there, those are snail embryos, and we scored the presence or absence of pigments by looking at the eyes of the snail. So you can see the darkly pigmented eyes on a couple of the snails, and then there's an albino in the middle, which lacks that pigmentation. So this experiment suggested to us that this is albinism in sort of the traditional sense, where a single gene is mutated that shuts off the production of the pigment melanin. There we go. There's an enlarged view of those cute little snail embryos, and you can see the dark eyes of the pigmented snails and the absence of pigmentation in the albino embryo. Now, I've had some excellent collaborators working with me on the focal pole snail cave project. One of those collaborators is pictured here. Her name is Helena Belanja. Helena is a Croatian biologist. She is a molecular biologist, and she runs a lab at an institute in Zagreb called the RiverWatch Institute. And Helena is the principal investigator in a program called Evolution in the Dark. So she's interested in what are the genetic and the molecular mechanisms behind the adaptations to darkness. And she and her students have adopted the focal pole cave snail as one of their model research organisms. So I'm going to share a little bit of their interesting findings with you. So this is a pathway, a metabolic pathway that shows us how that dark pigment melanin is produced. It is synthesized from a precursor amino acid called phenylalanine. So phenylalanine is converted into tyrosine. Tyrosine is converted into L-DOPA, and then that pathway continues into melanin production. And one of the findings in Helena's lab is that the albino form of the focal pole snail is albinistic because of a gene mutation that prevents the conversion of tyrosine into L-DOPA. And that has some really important downstream implications to these animals. It means that tyrosine can then be diverted into other metabolic pathways. And those include converting that tyrosine into dopamine and noradrenaline and adrenaline. And so if you remember your biology, those are really important hormones and chemical messengers. And so for the albino snails, they have elevated levels of dopamine and noradrenaline and adrenaline, which could represent an important adaptation for them for surviving in the relatively harsh environment of a cave. In addition to that, they found that the albino snails are experiencing a difference in gene expression. They found 742 genes that are either upregulated or downregulated in the albino form of the focal pole snail. So upregulation means the same genes are turned on more often, and downregulation means the same genes that are found in the pigmented snails are turned on less often. So these are genes that have to do with immunity and metabolism and sensory perception. So again, that shifting of that pathway away from melanin toward dopamine and adrenaline is part of this larger story of differences in gene expression in the albinos and the pigmented that, again, could represent important adaptations for helping them survive in the harsh environment of a cave. Now, here's a bonus critter for us. This is not a snail, but the snails are, of course, part of a biological community. And in some of the recent cave snail surveys, we have discovered that focal pole cave is home to a freshwater sponge, which was a pretty exciting discovery. So there's a freshwater sponge for you. It is growing on the underside of a rock in the cave stream. Next to it, you might recognize there's a little crinoid fossil. All right, so that's a little bit about the focal pole cave snails. The second cave and population of snails I want to talk about comes from Illinois Caverns. Illinois Caverns is also in Monroe County. It's owned by the Department of Natural Resources, and it is the only cave in the state that is open to visitation. So August through October, you can visit the cave and experience what a wild cave looks like. So this is a map of Illinois Caverns. You look on the left side of the screen, rather, you can see the entrance. There is a 5 to 6 miles of passage map. So it's less than half the size of Focal Pole Cave, but it also has a perennial stream, although it's small. So here's an interior view, maybe a couple hundred feet downstream of the entrance, and you can see there's a small perennial stream, and that stream is not only home to snails, but again, to a whole community of cave adapted organisms. Illinois Caverns is, again, ranks up there in this list of the most biodiverse caves in Illinois. There are 9 globally rare species and 9 troglodytic species known from Illinois Caverns. Here's the snail from Illinois Caverns. It is also a phycid, and it looks a lot like the albino morph from Illinois Caverns, especially the morph that has that sort of tan shell morphology. But you can see the lack of pigmentation. It has eyes, but those eyes lack pigmentation. So when we compare Illinois Caverns to Focal Pole, you can see that the caves are near each other, but they have distinct watershed boundaries. So effectively, we can think of these 2 caves as isolated systems, and the 2 populations likely have very little opportunity to interbreed. So we have the potential here that Illinois Caverns and Focal Pole snails are actually different species, albeit closely related. So one of the basic questions I wanted to answer is, are they reproductively compatible? So at least in the laboratory, the answer is yes. So this is a visual of an experiment where I took the albino snails from Illinois Caverns and paired them up with the pigmented snails from Focal Pole Cave. So by pairing them with the pigmented focal pole snails, we could use pigmentation as a marker. If they produce any pigmented offspring, if the albino Illinois cavern snail produced any pigmented offspring, we know that gene must have come from the focal pole side. And so you can see the intermediate form of that first generation offspring. And when they in turn produce the second generation, we get, again, a mixture of pigmented and albino individuals. So it's probably a very similar genetic mechanism responsible for the absence of pigmentation in the Illinois cavern snails as is seen in the Focal Pole Cave snail. So once again, my Croatian colleagues have stepped in and done some molecular biology on these animals. So this is a tree based on the sequence of the 16S ribosome RNA. So if you remember your high school biology, ribosomes are the structures in cells where proteins are synthesized. So it's a very important structure, and differences in the sequence of these molecules are really biologically significant. So if you pay attention to the red arrow there, you see that the Focal Pole Cave and the Illinois cavern snails actually form separate branches on this tree, so they are genetically distinct. And interestingly, they cluster with two other Phycella species, Johnsoni and Wright-eye. Those are aquatic snails that are found in springs in Canada, right? They're Canadian thermal spring snails, and they are genetically related to our cave snails here in Focal Pole Cave and Illinois caverns. Now, like is the case in Focal Pole Cave, the snails in Illinois caverns are part of a community of organisms. So again, for your river watchers, you will recognize there's some isopods here and there's some amphipods here. In the center of this picture is an Illinois cave amphipod. That is a federally endangered species. Illinois caverns is one of the type localities for it, and the cave snail is part of that community. Check out the isopods. The one on the right is a troglobitic, a stegobitic isopod. Look how long the antennae are and the cilia are at the back. They have no eyes, versus the one on the left. That's a spring-dwelling isopod that also lives in caves. So you'll notice much shorter antennae and less troglomorphic adaptations. All right, now the third cave that I want to talk about is Stemler Cave. Stemler Cave is in St. Clair County. And as Danielle mentioned, my wife Nancy and I are the current owners of Stemler Cave Nature Preserve. This cave entrance became a nature preserve back in 1997 when the then landowners, Homer and Loretta Stemler, enrolled it in the Illinois Nature Preserve's system. The Stemler family were the German immigrants who settled this area in the 1830s. And Johann George Stemler built a log cabin on site, which still stands today. And according to the Stemler family history, that cabin was built in 1836. This is what the entrance to Stemler Cave looks like in cross-section. This is a map drafted by members of the Illinois Speleological Survey. So you can see the entrance is a steep-sided sinkhole. It's immediately adjacent to Stemler Road. And when you enter the cave, if you turn around and look upstream, the cave roof has collapsed. You can't go upstream. If you turn and look downstream, the cave and the cave stream flows to the north. Now, Stemler Cave Nature Preserve should not be confused with Stemler Cave Woods Nature Preserve. Stemler Cave Woods is a Department of Natural Resource owned nature preserve. It's open to the public. You can access the hiking trail off Stemler Road. There is miles of hiking trail available there. There's the Homer Stemler Memorial hiking trail map. So it's a nice place, just like the Paul Whiteman Subterranean Nature Preserve. It's a nice place to walk through a karst landscape, see sinkholes, see small cave entrances, but you're supposed to stay on the trail. All right, so this map is showing stream systems. It's a GIS layer for surface streams. And if you look on the left side of the screen, you see that gap where there are no surface streams. That is the watershed for Stemler Cave. On the right side, in the red boundary, we see the actual delineation of the Stemler Cave watershed. So all the streams are underground in that area, and the spring there is the resurgence point for this cave. So the Stemler Cave has a very large recharge basin. It has a 7 square mile recharge area. So that's big, big. That large recharge area makes Stemler Cave very flood prone. It's collecting runoff from a 7 square mile area. So although the recharge area is about 7 miles long and 1 mile wide, there's only about 1.2 miles of main passage, and then another about 4/10 of a mile of side passage. So that means there's probably a lot of cave there that has yet to be discovered. There's certainly more cave at the downstream end, and there's certainly more cave at the upstream end, but it sumps full of water, and unless you're a cave diver, you can't really explore it. All right, so that map was created again by the Illinois Speleological Survey. I just want to give a shout out to them. It's a hardworking group of folks who map and document caves. This is what the spring looks like. This is where the water flowing through Stemler Cave surfaces. It's called Sparrow Spring, and Sparrow Spring forms Sparrow Creek, which flows on to Hickman Creek, and I believe eventually winds up merging with the Cahokia Canal. This is what the entrance to Stemler Cave looks like. You can see it during lower and higher flow conditions. In fact, on the right, we're talking about flood conditions. So after really heavy bouts of thunderstorms, the cave can completely pipe full of water. And look how much soil has been picked up, right? So it looks like chocolate milk. This is what the cave looks like looking downstream. So for the size of the watershed, the cave really isn't that big. If I was standing right here, I would be about knee deep in water. And if I stretch my arms out to the side, I might be able to just barely touch the side walls of the cave, although the ceiling would be probably about 15-20 feet high in this area. Because it's flood prone, the cave doesn't have very many delicate formations, but there are some pretty cool flowstone formations in the cave, like this large flowstone stalactite complex. We also have some big sections of breakdown. So here, this is Steve Taylor, who's a retired biologist from the Natural History Survey, sitting on a ledge next to a large chunk of breakdown. And behind him, you might notice a piece of orange flagging. That's a survey marker when the cave is being surveyed. Okay, now let's talk about the biology of Stemler Cave. Stemler Cave in that 2003 study ranked #2 in biodiversity among the sinkhole plain caves, 14 globally rare species, and 10 troglodytic species. So quite a biodiverse cave. I've been tracking the biodiversity of the cave since my wife and I moved here about 20 years ago. Adding to the published record or my own observations, there's been over 100 different species documented from the cave. When you look on this graph, check out the snails toward the bottom. 17 species of snails have been recorded in the cave. There are 2 of the species that I'm going to elaborate on here. Here's my bonus critter for Stemler Cave. A few years ago, I discovered hydra living in the cave. So this is a view through a microscope. Hydra are tiny freshwater relatives of corals and jellyfishes. They have stinging tentacles, and they feed mostly on microcrustaceans. So that was a pretty exciting addition to the known biodiversity of Stemler Cave was the hydra. All right, so the two snails that I want to focus on include this critter here. It's called the enigmatic cave snail, which I think is just a great name. This snail, unlike the physids, is a hydrobiid. So this would be a right-handed snail and an operculate snail. It's gill breathing, not lung breathing. They are very sensitive to water quality, and this creature is at the far end of that cave-adapted spectrum. No eyes, no pigmentation, and many of the life history features that we associate with cave adaptations. The species was originally discovered in Stemler Cave in 1940. It is also known in Cliff Cave over in St. Louis County and then down in Perry County. So in Illinois, it's an endangered species. Part of the enigma about this cave is its unusual geographic distribution. When we look at Monroe County, at all that karst area, and all those nice big caves with streams, there are no hydrobiid snails in any of those caves. And that's weird because in other areas where we have caves with streams, hydrobiids are the principal cave snail. So for some reason, the enigmatic cave snail never made its way to the caves in Monroe County. These organisms feed on biofilms mostly, right? So they're grazers. Think about a sheep grazing in a pasture. Well, these snails are grazing on films of bacteria and biological material that accumulate on the rocks, as well as probably things like leaves and debris that gets washed into the cave. This is Nicole Whitsitt. She was one of the Frick students and NGREC interns that helped me with doing some census work of cave snails. So that's one of the projects we've conducted is quadrat sampling to determine the population density of cave snails and which part of the caves they are found in. So these snails are tiny. An adult enigmatic cave snail is about 2.5 to 3.5 millimeters long. Nicole here is looking at a snail on that rock. I just highlighted it with the circle. So as you might guess, it's kind of tedious work to do in-cave research on such a tiny little creature. Here's some data from our cave snail census. So we find that the population density varies a lot through the cave. It's correlated with stream flow and substrate composition. We tend to find the snails in riffle areas with a lot of cobbles. We searched for them in the numerous side passages and found very few. So they really seem to be mainstream denizens of the cave. Now, in order to gain some more life history information about these snails, I took the approach of trying to rear them in the lab and do so under cave conditions. So I created my simulated cave in the lab using wine coolers and little snap top sort of Tupperware-like containers fitted with air stones and water that I collect from the cave, right? So I provided them with rocks and water from the cave and put them in a cooler and tried to let them do their thing, reproduce so I can gain some information about their biology. So this was successful. I managed to document their reproduction in the lab. This work was published. We see here a collage of images of the cave snail. Frame A there is a pretty newly laid egg. These snails lay their eggs one at a time. It's at the 4-cell stage, so that embryo's gone through 2 rounds of division. B is a well-developed embryo, and C is a new hatchling. When they hatch, they're about 0.75 millimeters long, and it takes them about 70 days to hatch. So this tiny little embryo takes 70 days on average to hatch. And then D there is an adult enigmatic cave snail. So not only did they hatch, but they grew in the lab, and I was able to document their growth rates, at least on a limited number of individuals, and man, are they slow growing. That is typical of stegobitic, of troglobitic species. They have low reproductive potential, slow embryonic development, and slow growth rates. It takes these snails essentially a year to reach the full mature adult size and even consider the prospects of becoming sexually mature. Here's another interesting, or at least to me, finding from this research. In the cave, these snails accumulate a heavy black patina on their shells. And that patina is probably made of the same minerals that blacken the rocks in the cave stream, iron oxide and manganese oxide. When I rear them in the lab, they lack that patina of minerals. They don't have that constant flow of mineral-rich water rushing over them. And their shells, you can see, are totally clear, whereas you might not know that in the field because of that heavy black patina. I was also able to rear the enigmatic cave snails from Cliff Cave over in Missouri. And I found some interesting differences in one of their behaviors, where they lay their eggs. The Stemler Cave snails almost exclusively deposit their eggs on the undersides of rocks, whereas the Cliff Cave snails predominantly lay their eggs on the top of the rocks. And there's some possible explanations for this. Two factors. One, the way Stemler has such a large recharge area and is so flood prone, we get these intense flood pulses, and by cryptically laying your eggs under the rocks, it would protect them. When you go to Cliff Cave, there's the water is totally saturated with calcium carbonate, and a lot of the rocks are cemented together. So the snails don't really have as much of an opportunity to get under the rocks. So they have perhaps this behavioral adaptation of laying their eggs on the top side of the rocks. Here's one more thing that I found interesting, that was a difference between Cliff Cave and Stemler Cave. Although these snails don't have eyes, at the base of their antennae, where their eyes would be, they have this patch of kind of iridescent cells. And in the Stemler Cave snails, that patch of iridescent cells is a milky white, but in the Cliff Cave snails, it's kind of an iridescent yellow. So I'm not really sure what's going on there, but it certainly represents some genetic differences between these two populations. Now, although Stemler Cave Nature Preserve is legally protected as a nature preserve, there are a number of threats to the ongoing existence of this cave community. Like the other caves in Southwestern Illinois, they're subject to nutrient enrichment. Those nutrients come from runoff from farms, golf courses, lawns, from septic systems. The Stemler Cave area is rapidly urbanizing. There's lots of new houses being built. There's erosion happening along Stemler Road that's introducing a lot of soil, and there are invasive species. So that's the second snail I want to talk about. It's an invasive species that is potentially competing with the enigmatic cave snail for the resources in the cave. So that invasive species is a phycid. It's called Physa acuta. It is known to be an invasive species in many surface habitats. And as far as I know, this is the only documentation of this snail invading a cave environment. So there is a reproducing population of Physa acuta snails in Stemler Cave. And the nutrient enrichment that's happening sets up this potential for non-cave adapted organisms to eke out a living in the cave, and in doing so outcompete the cave adapted organisms. So other projects I've done with students included raising the Physa acuta from Stemler Cave in the lab under simulated cave conditions, this is a little comparison of the data. With the Physa acuta, it only takes them about 8 weeks to go from a pretty new hatchling snail to adult size and starting to reproduce. In that same 8 or 9 week period, the enigmatic cave snail is just, a tiny little 1 or 1.25 long youngster. So they definitely have an advantage when it comes to their growth rate and their reproductive potential, setting up the possibility of competition between the two species. There's also undescribed species in Stemler Cave, and this is really true for most of the caves in the U.S. and in the world. Cave biology is behind surface biology. We have a lot of understudied and undescribed taxa, and there is a terrestrial snail in the genus Helicodiscus that has been documented in the cave that appears to be new to science. One of the other undescribed species in the cave is a springtail, and there's simply a shortage of taxonomic experts willing to take on the job of describing these undescribed species in cave environments. I'm wrapping up the talk here. There's lots of individuals who provided me with photos that I've used in this presentation. And if you're interested in learning more about this research, if you would like a bibliography of some of the published work I've mentioned, please email me at bob.weck@swick.edu, and I'd be happy to share that bibliography with you. And I can take on some questions if we have them.
Hi, Bob. It's Jeffrey Gosnell.
Yeah, Jeffrey, hi.
Hi. I'm wondering how a cave snail in Illinois has a genetic relationship to snails in Canada.
Well, yeah, they would share a common ancestor at some point, right? So if I can zip through my talk and get back to that slide here, I can focus on that again. So this is where are we at? This is a tree of relationships, right? So if we look at the Focal Pole Cave branch and you look at the Illinois Cavern branch and you follow that back, the point where they diverge was their common ancestor. And if you keep stepping back in that tree, just like a family tree, at some point there was a common ancestor that probably had a large geographic distribution, and the Canadian populations became isolated and diverged, and the Illinois populations became isolated and diverged, right? So at some point in the past, they share a common ancestor. Does that get at your question, Jeffrey?
Yes, thank you.
Okay.
And we've also have a couple of questions and comments in the chat that I'll share.
Okay.
Diane was excited to see the hydra. And
yeah, great. They're cool critters.
Yeah, she was wondering if you haven't seen any freshwater jellyfish in the caves.
No, I have not seen that. I've seen them in lakes in the Shawnee, Devil's Kitchen Lake, but no, not in a cave environment. In fact, Stemler is the only cave I've seen hydra.
So something that I've kind of wondered about is, you kind of briefly mentioned the different kind of riffle habitat and other habitats that were within the caves. Do a lot of the, with the diversity of different snails in the caves, do they have kind of preferential habitats of the riffle versus or the sediment type, or is it just kind of they're scattered in sharing all the different habitats?
Yeah, I think each taxon in a cave stream, just like in the surface stream, they have niches within the cave, they have habitats within the cave that they prefer. The physid snails seem to be more generally distributed, and the hydrobiids tend to be utilizing the riffle habitats more. And that would go true for different isopods and amphipods as well.
Right. Anyone else have questions? Again, feel free to type them in the chat if you'd like me to read it for you, or feel free to unmute yourself and ask your question aloud. All right. Well, I'll go ahead and ask something else that's maybe. Oh, go ahead if you have some.
Sorry. Hi, Bob. Joe Medley.
Hey,
We've
yeah.
met earlier.
Yeah. Joe's
I was
helped me with
curious-
the focal pole snail stuff. Yeah.
Yeah. So I was actually curious. This is probably a silly question, but when you actually measure the diversity in that, is that just basically species richness?
Yeah, by species richness, I just mean
Oh,
what's
no,
the taxonomic diversity?
Oh, did you say species richness in there? I did not catch that. Sorry. I was talking about the diversity measures when you actually look at the when you compare the diversity numbers between the caves.
Yeah.
Okay, that was species richness.
Yeah, it's really focused on the digobitic and sarclobitic species diversity. Yeah. Okay. So yeah, you could use species richness in the same way. Mm-hmm.
Okay. Yeah. Fair enough. Thank you very much.
Yeah.
So not specifically snail related, but do you have any particularly interesting or stories of your explorations in these caves, things that you think might kind of encapsulate what the experience of doing biology in a cave is like?
It's cold and wet and dark. Yeah, so you spend a lot of time squatting down with your butt in the water. That's why I tend to try to rear the animals in the lab, because it's just so much more convenient to do controlled observations when you're dry and warm. So yeah, there are challenges to doing cave biology. The caves in our area are very flood prone, so you have to constantly keep an eye on the weather, and lots of cave trips get canceled because of the threat of rain. So yeah, it is. There's mud everywhere. All your data sheets get covered with mud, and it's fun, but it's a hassle, right? You have to drag all your gear with you, and when you try to do photography, your lenses fog up and your glasses fog up, and yeah, there's a lot of challenges associated with doing cave biology. Yep. If you go into a cave with a biologist, it's like going on a hike with a nature lover. It's slow, right? So when you do cave biology, it's slow work. Yeah.
All right. Well, any last questions or final thoughts to share?
It's funny, actually. Sorry, I don't mean to take over. You say it's slow with the biology in the caves, and yeah, I'm a geomicrobiologist too. The science and resource manager over at Mammoth Cave, Rickard Toomey, when we went out there last time to do field work with them, someone had asked where his wife was, because she's normally out helping with the science, and he said, "Oh, she asked what kind of science it was," and I said biology, and she said,
she's
"No,
like, never
thank
mind."
you." Exactly. Thank you for the presentation.
You're welcome.
Yes, thank you very much for being here with us tonight, Bob.
Yeah, my pleasure.
Great presentation. And then also thanks to all of you for joining us tonight to hear about cave snails. Before you go, I wanted to mention that our next speaker series event will be on Tuesday, February 6th, again at 7 p.m. Manisha Pant from the Illinois River Biological Station will be talking about long-term stream monitoring that's been happening in Illinois that uses macroinvertebrates as bioindicators. And then also keep an eye out for our upcoming announcements of our spring training workshops. These will be happening in April and into the beginning of May. Workshop dates and locations will probably be announced in early February. You could register for the speaker series and the workshops and other future events using our webpage or our Facebook page. Once again, thank you very much, and I hope you have a great night.